Hello everyone and welcome to the next biology video. We're going to be looking at the eye and then vision problems and then hopefully an exam question as well. Okay, so there's quite a lot of information about the eye. So here is a cross section, and one of the things you have to be able to do is put on all of these labels. Okay. Retina, sclera, optic nerve, iris, ciliary muscles, suspensory ligaments, pupil, lens, and cornea. So if you want to, you can pause the video and just have a little go and see how many you can remember before we have a look at each of them. Okay, well done. So let's have a look at our labels and see if you got the correct names for the correct things. So a lot of information on here, and we've also got some explanations as well, but let's just look at the labels first. So the cornea is the transparent region at the front of the eye. Okay, so that was this label here. The lens is this oval shape here. The pupil is actually a hole, a bit like a stomata. It's actually a hole, the gap here, that's the pupil, and that's the black bit, the middle of your eye. And then the iris is the coloured bit, so it's this bit here and this bit here as well. Okay, now we've got two labels here which cover this, where am I, area, where were we, here, this area here. So the little circle is a muscle called the ciliary muscle, and the stringy bits there are called the suspensory ligaments. And it's really important we know which is which, because on the higher questions they ask us what happens to those when our lens changes shape and things like that. So make sure you know which of those is which. Now at the back here we've got our optic nerve. And that is a nerve, so it carries electrical impulses from the retina to the brain. And the retina is here, and this is the back surface of your eye. See if I can get my pen on it a little bit better. There you go. The back surface. Now, this contains rods and cones. And they are cells, but they're specialist cells, and they pick up light and they pick up colour. Now, cone cells pick up colour. Okay, so if you remember the C for cone, that's colour, and the rods, they pick up the light. Okay, so that's all of our labels. Now, let's have a look at their functions. So I've explained some as we go there. So we need to think about the retina, the sclera, the ciliary muscles and suspensory ligaments, and then some are going back the other way. So we've got the explanation and we need to put in the name. So pause the video for a minute and see how many of those you can fill in or get. All right, let's have a look at our answers. So you will need to know all of these, okay, for all of the different exam boards, foundation and higher. So, retina, light-sensitive receptor cells with rods and cones. Optic nerve carries impulses from the retina to the brain. Sclera is that tough outer layer of the eye, and it protects the front of the eye, and it also holds the eye in place as well. The cornea is the transparent region. So transparent means it lets light through. Right at the front of the eye, and it refracts the light. That means it bends the light. We'll look at how it does that in a minute. The iris is the coloured bit, and it has muscles to open and close your pupil. And if you don't know, when it's really bright, your pupil will close and get smaller so that not so much light gets in. And when it's quite dim light, it will open so that lots of light can get in. And then we've got our ciliary muscles and our suspensory ligaments, and they will change the shape of the lens so that the image focuses on the retina properly. Right, so well done if you've got all of those. Make sure you learn those because you will need them. And so let's have a look at the next bit. Okay, so in order for us to see something or focus an image, it's all about the lens here. Okay, this oval shape, this is our lens. And the thing about the lens is it can get shorter or fatter, or it can get longer or thinner. And we talk about how it changes in a second. But the general focusing an image is the light rays, we've got two of them, come into the eye. They slightly bend through the cornea, and then they bend again going through the lens. And what should happen, which it does on this one, is they meet bang on the retina, which means whatever you saw over here, let's say you saw a person, that image will focus on the retina. And then that image will be taken down the optic nerve to the brain where it's processed and you actually see that image. Obviously it all happens in milliseconds. It's not something we're conscious of. So key word here, I'm sure you've all heard this word in physics, but just in case you haven't, refraction. Bending of light, not reflection, but refraction. Okay, so you can see the light rays bend here, they're going in a straight line, then they bend. Okay, so that's general focusing, but let's have a look at when we look at close objects and when we look at far away objects, because the lens will actually change shape. And when it changes shape, it does that so that we can focus still on the retina. So let's have a look. Ooh, a couple of questions there. Right, so if we are looking a long way away in the distance, so a long way away, the light rays will be like the diagram we just seen. They will be coming in here parallel to each other because our object is over here somewhere. The only thing I can draw is little men, so, or ladies, sorry. They'll be coming in parallel, they're refracted, and then they meet on the retina, which is like the one before. Now, if we're looking at something near, it's slightly different. So let's imagine, what can we be looking at? I don't know. We're looking at a button. So it's close up, it's in our hand, and we're looking at this small object, and it's close up. The light rays, if you look, they're not parallel. They start at this single point, they diverge or go outwards, they hit the lens, and they're still focused on the retina, but it's this bit that is different for a near object. Now, it says here, what are the differences between the two images? Look at the light rays and the lens. So we've looked at the light rays, they're different. Now look at the lens. This one is thin lens. And this one has quite a fat lens, doesn't it? It's changed shape. We'll put wide lens. The reason being, if we had a thin lens here, this wouldn't focus on the retina, it would focus back here and we wouldn't see what we need to be seeing. So when we're looking at a distant object, the light rays are parallel, the lens is thin and long, and then we're looking at a near object, the light rays diverge and the lens is wider and shorter. Okay, so that's the difference between a distant object and a near object. Okay, This is basically the same diagram, just written in a slightly different way. Let's have a look at the lenses and the parallel lines. So distant object, parallel lines, thin lens, near object, divergent light rays here, and a thick lens. Now if you need to write it down rather than having to draw the pictures, I put this for you here. So if you want to pause and just jot it down, you could do that now. Right, well done. Let's have a look at the next one. Okay. Accommodation. Now, not accommodation as in your house or your flat. This is accommodation. This is just showing us how the lenses change shape. So let's have a look. I can get the my pen on the lens. Where is it? Oh, it's all the way over there. So this lens here is short and fat. And this lens here is long and thin. Okay. And accommodation, the definition is here. The process of changing the shape of the lens to focus on near or distant objects. So it's what we've done before, but we're just looking at a bit more detail. Now I said at the beginning you'd need ciliary muscles and suspensory ligaments. Okay. So if you're looking at something close by, so near vision, remember, looking at something close up, the lens from the previous slide is quite short and fat. So this is what we need to know. The ciliary muscles contract. The suspensory ligaments relax. The lens becomes thicker and rounder to focus on near objects. Now this is often a 6 marker, this, so we need to know these. And then when we're looking a long way away, distance vision, ciliary, exactly the opposite. Ciliary muscles relax, suspensory ligaments pull up. So in this one they relax, in this one they're pulling. And then the lens becomes flatter to focus on the distant object. Okay. Now, you only need to learn those 6 little lines that I've underlined. 1, 2, 3, 4, 5, 6. Because if you get a 6 marker and it says, describe how the lens changes shape looking at near and far objects, these are your key words that you're going to need to put in. Okay. Right, hopefully then you're all okay with accommodation, but if you need to jump back and have another look, that's fine. Now this is the same as the slide before, but I've just put it into a table for you. So, so it's a little bit easier. I'll just move myself out the way so that you can see the box. There. So if you want to jot down what we've just said, but in a table form, pause the video now and you can jot that down. Okay, right, very well done. So now we know how it focuses and how we see objects. Let's look at problems with the eyes. Now this is me, long-sighted, hyperopia. Okay, big word, but hyperopia, long-sighted. Now, people who are long-sighted can focus a long way away, so they can see a long way away, but they can't see things close up, and that's me. I can look out the window and see something 500 meters away, but if I try and read my book, I need my glasses on, okay? So what should happen when we're looking at something a long way away, as our diagram shows here, if I can get my pen up, where's it gone? There it is. When I look at something a long way away, it focuses on the retina, it's fine. But if I look at something close up, what happens is it focuses here behind the retina. So it focuses too far back, it's too long. So long-sighted is behind the retina. Okay, and that's why people who are long-sighted, they can see a long way away, but they can't see close up so well. So that's long sight. That's rarer than short sight. So let's have a look at, oh sorry, long-sightedness can be caused by, so the lens can be too thin, or the eyeball is too short. Okay, or it could be a mixture of both. Because if the lens was thicker, here, it would refract the light more, it would bend it more, and it would end up here. Or if the eyeball was longer, the retina would be further back. Okay, so that's long sight. And short sight is the opposite. So short-sighted people, they can look close up, they can read a book, and when they're looking close up, the image is focused on the retina here. But when they look a long way away, because it's short-sighted, it doesn't go far back enough. Look, it's too short. Sorry, pen's having a moment. Where are we? There we are. It's too short. It should be back here, but it's here where it focuses. And again, oppositely, the reason it can be caused by the lens being too thick, so it refracts the light too much here, or the eyeball being too long or too big. Because if that eyeball was shorter, it would actually focus on here, but the back of the eyeball would be there if it was shorter. Okay, so we've got long-sighted hyperopia, short-sighted myopia, and the reasons why they happen. Right, now if we want to correct these things, we can use lenses. So she pushing her glasses up her nose. Okay, so we've got three, well actually there's four ways. So wearing glasses is the most common one. And what the lenses here actually do is help that eye focus on the retina instead of in front of it or behind it. And two different types of lenses here. So we've got a concave lens for short-sighted and a convex lens for long-sighted. Or we can have contact lenses. So they work the same way, but they just sit on your eyeball rather than being in front. Now, I'm just going to move my picture down so I can put on the lenses. So a convex lens, this one, is, oh, try and get my pen back, is the oval shaped one. And the concave lens is that shape. So I've got the convex lenses because I'm long-sighted. Right, so this is how we can correct vision problems. There's also surgery these days where they can do something with your lens, I believe, and do like a permanent solution, but it's not that common. You can pay to have it done. But for your exams, these are the four things that correct vision problems. Okay, so hopefully they all make sense. There it is, eye surgery. So they change the shape of the cornea, so that transparent bit at the front, they change the shape of that so that the eye can focus directly onto the retina. You do have to have local anaesthetic. So local anaesthetic, you're not asleep, but they will make sure that nothing hurts. I'm not sure about trying to keep your eyes open while someone's working on it, but lots of people have it done. Right, so there's our sight problems. And now we've got an exam question. So I've just done one this time. So some people wear glasses to improve their vision. Shows light entering the eye in a person with blurred vision. And it shows how this condition is corrected with glasses. Explain how the blurred vision is corrected. 2 marks. So if you want to pause and have a go, please do. Right, we're going to have a go now. So explain how the blurred vision is corrected. So the first thing we're going to say is either they're short-sighted or the image is focused in front of the retina. Okay. So image, real trouble with my pen tonight. Focuses in front of the retina. So we have a concave lens. Ooh. And that concave lens diverges the light slightly. So the image is on the retina. So key words here. It throws in front of retina. You could say, because they're short-sighted, you could say myopia. And then a concave lens diverges the light. So the image is on the retina. That's our key words. All right, remember, biology words get biology prizes. So very well done. Rewatch the video if you need to write anything down, and I will see you all on the next one. Take care, everyone.